available and where they can be applied. So I think then we can go to the third speaker of today's webinar, which is Dr. Serbulent Unsal, who is a senior machine learning engineer at Antiverse with an affiliation at Karadeniz Technical University in Turkey. Dr. Unsal applies AI methods to improve therapeutic antibody design with research projects in cancer biology, immunotherapy, and bioinformatics. And we look very much forward to hearing your insights. Doctor, the floor is yours.
Thank you, Erwin. Today, my presentation is more focused on application side of the things, so how we apply AI methods in the industry. So let's start the show. Yeah. First, I'd like to introduce the company that I'm working with, which we design therapeutic antibodies. Antiverse is an artificial intelligence-driven tech bio company specialized in antibody design, especially against challenging targets, including GPCRs and ion channels. We are headquartered in Cardiff and have offices in Boston. And also some branches in Canada and Japan as well. So we try to pioneer AI-driven biological discovery, especially for challenging targets. The main motivation behind this is you can still design antibodies for soluble proteins, even if it takes time. But for challenging target, especially for membrane proteins like GPCRs and ion channels, the success rates in the design campaigns are quite low. So in the current situation, we have more than 60 plus projects against 9 active targets program. And some of these targets are for some of these targets, we are collaborating with top 20 pharma, and we could issue 1 in 10,000 AI binder prediction accuracy, which the total program for each target takes at most 6 months. And 16 structure predictions each second is possible with our in-house models. So the AI, although we defined ourselves as an AI-aided therapeutic antibody discovery company, machine learning or AI itself isn't enough for itself to create a successful campaign against a target. Also, a tight collaboration with laboratory with wet lab is quite important. For example, without hyperexpression cell lines, we couldn't able to get successful results, or we couldn't able to try our results quickly. Also, without successful sequencing, we couldn't apply multi-parameter clustering to select right clones. So, in an end-to-end manner, this pipeline shows how our approach is working. When we have a target sequence, we start with stable cell line development, which means with 1 million receptors should be expressed on the cell surface for a successful campaign. Then we apply our machine learning and AI capabilities, which we have sequence-based and structure-based models to design our binders. Each type of models has its own pros and cons. Sequence-based models are quite fast and resource effective and cost effective as well. You can generate humanized antibodies or human-like antibodies with generative sequence-based models, then you can score them for binding against a specific target or against a specific domain. This is very, as I said, cost effective, but the prediction rate or success rate is quite lower than structure-based approaches. In the structure-based approaches, although they have very high computational costs, once you define your epitope and the pose of binding, these models could generate alternative structures for you, whether these might be paratope specific in terms of antibody CDR specific mostly, and epitope specific at the same time. However, for these models, the number of generated structures and sequences are limited, but still they need to be filtered when you are creating a library. In this one, still you can use sequence-based models to select best possible binders from these from these structure-based models. So you can combine two as well. Also, the affinity maturation and for developability, there are AI-based models exist as well. Some are in-house, some are public models, but we also do filtering based on more manual approaches like searching liability patterns. And finally, when you are creating your library, you need to convert your amino
I'm sorry, is it just me where the
I think
presentation
he's
is?
connected.
I also have a problem.
Oh, okay. Yeah, that's a bit unfortunate.
Erwin, can you hear me?
Yes, I can hear you. Yes.
I think Serbulent, he was disconnected, so
He's out of the chat. Let's
yeah,
wait for a few minutes
he's disconnected. Yeah.
to see if he comes back. In the meantime, let me turn on my camera. In the meantime, because we have to make sure we're not taking it too long this time, we can address a question maybe while waiting for him to come back.
One second.
There was I asked a first question about, I will read it for everyone. So, semi-mechanistic pharmacometrics models are widely used these days in drug development, and the concept of model-informed drug development is widely accepted also by regulators. But what about AI? Do you think that in 10 years from now we will talk about AI-informed drug development? But I see. Okay, we're back. We're back. Okay, then we'll keep the question for afterwards.
I had a networking issue.
Happy that you're back. You can share the screen and continue your presentation.
Yes, very much. Yes. I'm about to say, after designing libraries with AI, we do biopanning with page display in multiple rounds to find our binders. Then do NGS, and from this NGS data, we use multiple parameter AI clustering for binder selection and design. Afterwards, the expression of maps and testing for functional tests, competition assays, or affinity or selectivity and developability is the last steps we are applying. Okay. Another reason why we select the GPCRs, which I mentioned in the beginning, is now there are more than 400 GPCRs and ion channels associated with third-class disease exist. However, there are only three antibodies, three FDA-approved antibodies is on the market. So it's a very challenging target to aim. But we think that AI will play an important role in success against these targets. Okay. And as I said, we are collaborating tightly with our lab team. And here, other than AI, having cells with high receptor density is quite important here. I mentioned that we are using a structure-based antibody design to make epitope-specific binders. So we also do use more classical methods like molecular dynamics simulation and clustering conformational repertoires for of these target structure, because actually when you have a target you get a structure on PDB, but it's just one of the conformations which you can observe in reality. More classical methods like molecular dynamics simulations really helps here to us when designing epitope-specific antibodies, because one epitope might have multiple conformations, and we are basically designing against all these alternative conformations, which increases our success rate and performance. Also, we screen generated antibodies for stability, humanness, and likelihood of binding through unproductive antibodies, resulting in a high quality de novo epitope specific binders, which we synthesize and do the functional testing afterwards. Shortly, I'll show some more results, but with all these, we achieved to design epitope-specific libraries against more than one challenging targets. And also, the clustering is important because once you get the NGS results, you are ending up with many similar sequences. And with a multi-parameter clustering, you can select a diverse set of binders, because we know that not all binders will be functional at the end of the day, and having functional efficacy is our ultimate goal to put these antibodies into a patient. Also, we have many structural models, and the origami is one of them, which we use to predict antibody structures in a high throughput manner, which we need fast prediction and accurate prediction, because we will end up with hundreds of antibodies when you are starting design library, and at the end of the library you will have millions of antibodies. So high throughput antibody structure prediction is one of the key here. For this model, we are basically incorporating multiple other models, such as graph neural network, large language models, and also prediction of direct atomic coordinates afterwards without using of Rosetta. And on a single machine, a 100 GPU, this model can predict 16 structures per second, which means like 1.3 million structures per day, and it could be easily scaled. And until now, with all this technology, we achieved to complete 4 successful programs over 18 months, 2 GPCR, 1 T-cell receptor, and a transmembrane receptor. And these are, in these ones, we show ex vivo efficacy, domain-specific design success, also cross-reactive antibodies, which AI and clustering technology.
It's right, we have the same.
I think he lost the connection again.
Yeah, that's a bit unfortunate. So I don't know, Mehmet, what you suggest. One other option is also to
What is that?
he's out to ask him to record the presentation that we can then share it on the website. Then it will also be in better quality.
So we can have like the questions for like the first 2 presentations.
Yeah. Okay, let's begin on the question I just asked. I don't know, Nicola, Mehmet, what do you think?
Well, I think we will probably talk about artificial intelligence informed, let's say, dosing, but also there is options you mentioned. Probably will not only quantity, but also quality of data. How do we enable those data and the noise of data? It is not. It can be based on the collaboration with other. We can enlarge data sets, but there is also much of the noise that can be captured. You talk about artificial intelligence in informed dosing, but also I think this will be more a combination of pharmacometrics and artificial intelligence. But in the first years, let's say maybe in the next 10 years, but it will be also combination. I'm not sure it will be only AI.
You will tell, yeah, indeed.
And also there is for the second question, there are initiatives like I think European Union have some initiatives. Also FDA machine learning good practice. Also I think some academics also have some initiatives regarding the data. Basically, we will need, in order to provide some reliable models that can be used, we need standardization with the data set. So some guidelines on that standardization, which will be acceptable by all stakeholders, let's say like that.
Yeah. Nicola, I see Serbulent is back. Let's give it one more try and see if we can finish it.
I'm very sorry for these network issues. I try to sum up quickly for the sake of the time.
Okay, no worries. Thank you.
Okay, good. Yes, let's try to be more quicker. As I said, you can see results of this domain-specific antibody design campaign with AI. These signals are murine and human, and we have more than 8 successful binders against human and 2 successful binders against both murine and human in this campaign. Also, these results are, as I said, both validated with ELISA and flow cytometry as well. And also, we can say that we can compete against known binders in these targets, and we can also show functional efficacy against some of these targets, and some for the others, functional tests are still going on. And yes, you can see the thrombin-stimulated generation and the antagonism percentage here. And also we can do human platelet aggregation inhibition test for the target PAR1, which we have patented our results, our antibodies against this. And you can see it takes time to design a campaign and getting functional data, but we have one target at preclinical stage and one target at the beginning of the preclinical stage. And there are other targets against different indications such as oncology, IBD, thrombosis, and neuromuscular diseases against a variety of different target class. And we are partnering with, of course, CROs and big pharma companies to do these campaigns. And this is our team. Murat and Ben are our co-founders. Gersi is head of lab, and we have a pretty wide set of advisors and consultants as well. So I think I'm a bit over time, but I try to summarize the efforts and try to show how we use and what are the effects and results of use of AI in the industry for therapeutic antibody design.
Well, thank you very much. It was really nice to also have your perspective here today. We had, I think, 3 excellent lectures showing all the different techniques that we have, and also the diversity of all the applications. I think it's really impressive of all the opportunities we have, and I think the future will tell us what will be realized. Are there any questions from the audience? Feel free to turn on your mic and to ask the question if you want, or to drop it in the chat. There are no questions. Maybe I'll ask a quick one just to keep it short because we're already going over time. A small question is because now we've heard all the opportunities and all the positive stories that we will hopefully realize. But of course, there's also some risks associated to it. And then one of the things is that these AI algorithms can be biased. And when talking about healthcare, that they might result in disadvantaged, in poor predictions for certain outliers or vulnerable patients, thinking about socioeconomic status, race, ethnicity, sex, gender. So is any one of you aware of any initiatives to contain these risks, or do you have anything to share on that topic? I don't know. I think, Nicola, you already mentioned before, maybe Mehmet or Serbulent, are you aware of?
It's not basically. I think the downside of this is this type of biases are studied, but mostly by AI ethicists or healthcare ethics experts, not actively monitored. For example, by the journals, by the editors, or by the regulatory bodies. So, but I think we still in an explorative phase of AI, and during time, I hope more explorative bodies are aware about this. For example, we could easily kill a drug program because of the side effects, but these side effects might be identified or might be observed only a set of patients which has specific mutations or who has specific type of molecular profile. But even there is no way to show this to regulatory bodies and say that, okay, our drug is not that bad. Just we need to exclude this type of patients based on their molecular profiles, because all these things are very new, and even in, for example, patents, still regulatory bodies doesn't have too much experts who could understand the methodology when you do drug design using AI. So, but I think it's the initiatives like cost actions and reports conducted by them will be very helpful to create awareness in these topics.
Yeah, I fully agree. I think, yeah, absolutely, I fully agree. And I think also what you mentioned, the communication is very important. It's a new tool. We've all heard about it. And some people are just probably too scared. And that doesn't mean that the risks are not there. The risks are real. But I think education and then is very important. And yeah, communication is important. Thank you. Thank you all for contributing to the webinar today. I think it's time to wrap it up for this year. But Enota will keep going on, of course, in the following years. So you all know the website, enota.eu. Please keep a look at the website. We will communicate also new initiatives in the following year. So it was a pleasure to have you here today. Thank you all, and I wish you all a very nice end of the year.
Thank you.
Thank you all.
Bye-bye.
Bye-bye.